The life of the cell from when it's first made to when it divides is called the cell cycle. And again, it's a series of phases that lead to cell division. The phases are called G1, S, G2, and M. G0 is an alternative that some cells will go into if they're not going to divide. So this is what the cell cycle looks like. So this is when a cell is first born, if you will. So when a cell is first made. It goes into G1 first. It's called the first gap. This is a period of growth. At a certain point, it'll reach a point where it decides that it's definitely grown enough, it's definitely in a good position to continue into the next phase, which is the S phase. S stands for synthesis, for DNA synthesis. And so we can see in this picture that I have a single chromosome. So I'm going to focus on this one. And you see here that now I have two sister chromatids. So a chromosome is copied and becomes two sister chromatids. And that happens to each chromosome that was present in the cell. So S is for DNA synthesis. Then the cell will go into G2, or what's called the second gap. And again, this is a period of growth. The cell will be making proteins, getting bigger. If all goes well during that stage of the cycle, it will continue into the M phase. This is where the genetic material will actually get divided. So you can see the chromosomes here. The chromosomes will line up, and we're going to talk about this process in great detail. And then these chromosomes are going to get split. And then the second piece of this is cytokinesis. Cytokinesis is when we actually get splitting of the cytoplasm. So mitosis divides up the DNA. Cytokinesis divides up the two cells. So we're going to go through the cell cycle a little bit more detail here. During G1, the cell is going to decide to divide. So it's going to be looking for certain signals in the environment and within itself to decide if it's in a good position to divide. If it's in a good position to divide, it will pass through something called the restriction point. This is a place where the cell basically has to stop. It's like a stop sign. And the cell has to look both ways, this is kind of how I think of it, and see, do I have the materials that I need to divide? Are there the resources I need to divide? And is there signals in the environment and within myself that I should divide? If the answer to all of those questions is yes, then the cell will continue into the next phase, the S phase. The S phase is when the chromosomes are copied. So remember, S stands for synthesis. So this will be when sister chromatids form. So again, sister chromatids are when you go from having a single chromosome to having two identical copies of the chromosomes. And they're still held together. They're held together in this location called the centromere. So here it is on this one. And this area is going to be important because in a later stage, that's going to be an attachment site. So during the M phase of the cell cycle, this will be an important piece of it. And then notice that there's an extra thing being shown here. So we went from the first chromosome to the sister chromatids, and then it goes to looking like this. This is what's going to happen at the beginning of mitosis. So this is mitosis, the later thing that we're going to talk about, or the later phase rather. This is what happens at S phase, which is what we're talking about right now. So S phase is going to take this chromosome and copy it into two sister chromatids. Later on in the mitotic phase, the chromosomes will become really, really tight and make this specific kind of shape. So people often get confused with these terms sister chromatids versus homologous chromosomes. So remember that homologous chromosomes are ones that belong in a pair with each other. So they should match each other. And the best way to tell if they match is to look for the same size. So if we look at this image here, the homologous chromosomes are going to match each other. So this one matches this one matches this one, and this one matches this one. So this is a homologous pair, that's a homologous pair, and this is a homologous pair. Sister chromatids are exact copies, not just similar, but exact copies. And so the sister chromatids are the two ones that are connected. So these two are sister chromatids, these two are sister chromatids. These are sister chromatids, these are sister chromatids, these are sister chromatids. So if I'm looking at this picture, how many chromosomes are there? There's 1, 2, 3, 4, 5, 6. So there's only 6 chromosomes present here. How many chromatids are there? Well, each one of the chromosomes is made up of 2 sister chromatids right now. So this is actually 12 chromatids. But again, because these two are just identical copies of each other, that's only representing 1 chromosome. Okay, so now we're going to talk about the rest of the cell cycle. So let me just kind of took a detour there for a second. So we were talking about G1. We then said we went to S, so that's why we made those sister chromatids. And once we've made the sister chromatids, the cell goes into G2. So this is another time for growth where the cell is making proteins that it's going to need later on. And then if it's able to do that and progress through that normally, it will go into the M phase. This is when the cell will actually divide. So in the first part of it, mitosis, it will divide one cell nucleus into 2. So this is dividing up the DNA. Cytokinesis is dividing up the cytoplasm. So this is what will normally lead to 2 new cells. So I get 2 new nuclei from mitosis, and cytokinesis gives me 2 new cytoplasms. Mitosis, the M phase. So here's the whole cell cycle, G1, S, G2, and the M phase. The M phase is broken into multiple parts. So again, we said mitosis and cytokinesis, but within mitosis, we also have 5 steps: prophase, prometaphase, metaphase, anaphase, and telophase. And it's this sequence of steps that's going to divide the nucleus. And then again, the cytoplasm is getting divided as cytokinesis, and that's what will actually lead to the 2 new cells. And then these 2 new cells will progress and now start over in G1. So when we say that cells are born, that means they just came out of finishing the regular cell cycle. Now earlier I mentioned G0, and so I just want to draw onto here G0. So G0 is when cells leave the cell cycle. Cells that are in G0 are not dividing. So these are cells that, the ones that probably are most well known are things like your spinal cord cells. Spinal cord cells are in G0 permanently, so they can't divide. So if I kill a spinal cord cell, so imagine I have a spinal cord injury and that kills the spinal cord cell, the other spinal cord cells, they can't go through this cell cycle, so they can't divide. They're all stuck here. So when cells are in G0, they won't divide. Some cells are permanently in G0, like the spinal cord cells. Some cells will only temporarily be in G0, and they can be stimulated to leave G0, so and then rejoin the cell cycle and divide. It depends on the type of cell. So why does a cell divide or not divide? And also, how fast does it divide? So there's this decision: should the cell divide, and how fast should it divide? And the cell is going to rely on both external and internal signals to determine that. So both external and internal signals are needed to make this decision. External signals, the environment plays a big role in that, especially in unicellular organisms. So things like bacteria or yeast or other things that are unicellular, their decisions about division are going to rely on just like how much food is in the environment. Multicellular organisms like us are going to rely on signaling. So remember signaling, there's a signal that's sent out and other cells receive it and have a response based on it. So this could be the bloodstream, this could be the extracellular matrix of your tissues where these signaling molecules are occurring. And there's a specific family of signaling molecules in animals called growth factors. So they get that name growth factors because they stimulate growth. Plants have similar ones called cytokinins, and same idea. Those signals, if they're present, will stimulate cells to divide. A classic growth factor in animals is called epidermal growth factor. You can catch here that epidermal growth factor was first discovered in skin. It's secreted by endocrine cells, so this is a long-distance signal that then causes skin cells as well as other cells to divide, but classically skin cells was where it was first discovered. So they're going to increase many of the genes that lead to cell division, and so they're going to not just cause a cell to decide to divide, but make it be faster. So when EGF is present, cells will divide faster. So these have normal functions in our body, and of course, like with skin cells, we always want them to be dividing and replenishing themselves. But if you end up with problems in your growth factors, so mutations in the genes that control these, so for example, in the gene for EGF, it can cause there to be too much growth factors or too much signal. And so this will lead to too much division. And too much division, cell division, is really one of the hallmarks of cancer. So many of these growth factors have been connected to different types of cancer. So there's a growth factor there's multiple growth factors actually that are important in breast cancer, in prostate cancer, in skin cancer. So these growth factors will have problems that contribute to many of the cancers that people get. So the cell is not just responding to its environment. So I said that external signals control division, but there's also an internal rhythm. And I like the word rhythm because it's almost like an internal clock. It uses proteins to make this internal clock. So basically it takes time to make proteins. Right? So the cell has to transcribe and translate that. And so if I have an increasing amount of the protein, that's going to tell the cell that more time has passed. Right? So that's why it's like an internal clock. It takes time to make the protein. So as the protein levels rise, the cell knows that more time has passed. It relies on a specific type of protein to do this. It's called a cyclin. So cyclin levels will rise as a measure of time for the cell. And so only when cyclin levels are high enough will the cell be able to progress. So if I give you a graph here, this is cyclin levels. Sorry, let me rewrite that here. Cyclin levels on the y-axis. This is the cell cycle on the x-axis. And so this is G1, S, G2, and M. So what happens is throughout G1, the cyclin levels rise, and they peak right before S, and then they drop. And then they rise again, and they peak right around the middle of G2, and then they drop again. And so the cell uses those rising levels of cyclin, again, to track time, to help it know that it should go into the next stage. So it won't go into S until it reaches this peak. It won't go into M until it reaches this peak. So again, this is a way for the cell to have some sort of internal clock for whether it's ready to divide or not, regardless of what the environment is saying. So the last note about the cell cycle is that it has these checkpoints that are critical to how fast the cell cycle progresses. I already talked about one of them. This is the most important one. It's called the restriction point. And so remember that a checkpoint is like a stop sign. So the cell is walking along through G1, and it's going to hit the restriction point. It has to check that everything's okay. And in this case, it's checking for the right signals in the environment in particular, and for cyclin levels. So it's checking environment and those cyclin levels. And if everything's okay, then it will go into the S phase. It will copy the DNA, and then it will go into G2. And then during G2, it will hit another one of these stop points. Oh, my stop sign's getting a little messy here. It's going to hit a different stop sign. And at that stop sign, it's going to check for environment, it's going to check the cyclin levels, and it's going to check another thing, DNA. So it's going to make sure the DNA hasn't been damaged. Why is this a good time to check for DNA damage? Well, remember, in S phase, I was messing around with my DNA. I was copying it. So that was an opportunity for a mistake to happen in the DNA. So there'll be a check for DNA damage at the G2 checkpoint. And then if everything's okay, it will move past that checkpoint into M and divide. But there's also an additional stop in mitosis called the Metaphase Checkpoint, and this will check that chromosomes are attached properly. Because during the M phase, the chromosomes are going to get divided. If they're not attached properly, they're not going to be able to be divided. So at each one of these checkpoints, the cell will stop and delay the cell cycle and try and fix the problem. If the cell can't fix the problem, it won't divide and will eventually die without dividing. So these checkpoints are really an important part of protecting our bodies and our cells from passing on problems or mistakes, especially with the DNA or the chromosomes. We don't want mistakes to get perpetuated in later cells, so those mistakes need to be caught here. And so that's how our cells help to make sure problems don't happen in later cells. So we'll wrap up this part of the videos with a couple of questions. So starting with the fertilized egg, a series of 5 cell divisions would produce an early embryo with how many cells? So if you think about that for a second, most people will jump to this answer. And that is not the right answer. So let me walk you through this one. So if you have one cell, it's going to divide and produce 2 cells. This cell no longer exists when those divide. Now these both divide. These no longer exist. So this is one division. This is a second round of division. Now these divide. This is the third round of division. We draw on those cells. Then these divide again. This is the fourth round of division. Draw on my cells here. And then now all of these guys are going to divide. Make sure I don't miss any of them. So this is my 5th round of division, and draw on my cells here. Okay, I'm just going to go across. That's taking forever. Okay, so at each one of these there's a dot. And if I counted those, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32. So after 5 rounds of cell division, I would have 32 cells. The easier way to do that is to do 2 to the n, where n is the number of divisions. So 2 to the 5th power, because there was 5 cell divisions, gets me 32. So that's a lot more than 10. So that's because every cell is dividing. And so this is exponential growth, and that's why the zygote, the fertilized egg, will become many cells very rapidly. The next question here is, how many chromatids are in a duplicated homologous pair of chromosomes? So let me draw that one out for you. I've got a homologous pair. And when they're duplicated, that means they're copied. So now I'm going to have 2 copies. Whoops, sorry about that. Now I'm going to have 2 copies of the red one and 2 copies of the blue one. So that's still a homologous pair, but they're copied. And so this is 2 chromatids and this is 2 chromatids. So there's a total of 4 chromatids in a duplicated homologous pair. The last question is, again, looking at some of these terms. A chicken has 78 chromosomes in its somatic cells. So remember back to the mutation chapter. Somatic cells are our body cells. And the question is, how many chromosomes did the chicken inherit from each parent? Well, remember, if we're a diploid organism, we get half of our or sexually reproducing organism, we get half of our chromosomes from one parent and half from the other parent. So this chicken must have gotten, so I'm just dividing 78 by 2. It must have gotten 39 chromosomes from each parent. 39 from the hen, 39 from the rooster gives you 78 chromosomes. How many chromosomes are in each of the chicken's gametes? Well, remember that gametes are eggs and sperm. So that means there must have been 39 in the eggs and 39 in sperm. How many chromosomes will be in each somatic cell of the chicken's offspring? Well, again, remember that somatic cell means body cell, so that organism, its offspring, should get back to having 78 because an egg and a sperm will get together. Okay, last one. How many chromosomes are in a set? Well, remember that diploid organisms have 2 sets, so that means there must be 39 in each set. 2 sets of 39 would give you 78. Okay, so that's some practice with chromosomes.